1 5-gallon bucket, 1 pound of plain potato, a handful of forest soil, a small scoop of sea salt, and 5 minutes of your time. That is everything you need to start a biological factory in your backyard that doubles itself every 48 hours, that produces 6 different plant hormones simultaneously, and that outperforms the synthetic biostimulant products sitting on garden center shelves at $30, $40, sometimes $50 a bottle. The agricultural chemistry industry calls these compounds plant growth regulators. Scientists call them phytohormones. The Korean farmers who figured out how to brew them in a bucket 60 years ago called them indigenous microorganisms. By the end of this video, you will know the exact recipe. You will understand the molecular biology of why it works, and you will understand why the $4.5 billion plant growth regulator industry would prefer you never learn the name of the Korean man who gave this technology away for free. But before any of that makes sense, you need to understand what is actually happening inside that bucket. Because what we are talking about is not fertilizer. What we are talking about is a self-replicating colony of microscopic organisms that produce on their own the same hormones that plant breeders manufacture in industrial laboratories. The same hormones that control whether a seed germinates, whether a stem stretches, whether a root branches, whether a fruit swells, all produced by bacteria, all produced for free, all doubling every 48 hours inside a plastic bucket in your garage. If you have ever stood in the gardening aisle of a big box store, staring at bottles labeled with terms like microbial inoculant, mycorrhizal blend, soil probiotic, biostimulant complex, and wondered whether any of it actually works, you're about to learn something that changes the entire equation, because the substance inside those bottles is not a modern invention either. It is not a breakthrough of agricultural biotechnology. The microbes you're paying for were already living in the forest floor 100 meters from where you grew up. They are living in the soil under every healthy tree on Earth. And for thousands of years, traditional farming cultures across multiple continents figured out how to collect them, multiply them, and feed them to their crops using nothing more than rainwater, a starch source, and patience. The chemical companies figured out how to freeze dry them, package them, and charge you $60 for a teaspoon of what you could grow yourself in unlimited quantity. What you are about to learn changes how you think about soil, fertility, and the products lining every garden center shelf. Because the story of microbial agriculture is not just a story about bacteria. It is a story about how indigenous farming knowledge gets erased, how simple biological processes get monetized, and how a billion-dollar industry rises on the back of organisms that any farmer could cultivate in a bucket for the price of a potato. This story begins, in its modern form, in 1965 in a small farming village in Suwon, South Korea. A young farmer named Cho-han Kyu had been watching his neighbors slowly destroy their land. Korea in the 1960s was in the middle of a green revolution. Synthetic fertilizers from Japan and Western chemical companies were being pushed onto Korean farmers as the future of agriculture. Yields rose in the first few years. Then the soil began to fail. Crops became dependent on ever larger doses of chemical input. Soil organisms vanished. Plants weakened. Disease pressure increased. The farms that had fed the same families for 15 generations began to require imported chemicals every season just to produce a harvest. Cho-han Kyu refused to participate. Instead of buying what the chemical companies were selling, he turned his attention to something he had watched as a child. Underneath the leaf litter of any undisturbed Korean forest, the soil was alive. It was loose. It was dark. It was teeming with white threads of fungal hyphs and microscopic life. No one had ever poured fertilizer onto that forest floor. Yet trees grew there for centuries without intervention, building their own fertility from nothing but rain, sunlight, fallen leaves, and the activity of the microbes beneath the surface. Cho Han Kyu spent the next 30 years asking a single devastating question. If the forest could fertilize itself for free, why could not a farm do the same? The methods he developed became known as Korean natural farming, or KNF. The central insight was this: healthy soil is not a mineral substrate. Healthy soil is a living ecosystem dominated by microorganisms. If you collect those microorganisms from a healthy forest floor, multiply them under controlled conditions, and introduce them into agricultural soil, you can regenerate the same fertility that built the forest in the first place. He called these collected microbes IMO, indigenous microorganisms. He developed a 5-stage process to capture them, feed them, and stabilize them for application. The technique spread quietly across Korea throughout the 70s and 80s. By the 1990s, his son, Cho Young-song, had built on this work to create a simpler, faster, more scalable version. He called it Jadam, a Korean acronym that translates roughly to people who resemble nature. The flagship product of the Jadam system is JMS, Jadam Microbial Solution, where his father's IMO required a 5-step process spanning weeks. Cho Young-song demonstrated that a similar microbial population could be cultured in a single bucket in 2 to 3 days. He gave it away. He never patented it. He published the recipe in books and on a free website now used by over 100,000 farmers across Korea, Africa, Asia, and the Americas. The Jadam website, currently maintained at n.jadam.kr, contains lecture videos, recipes, and field trial data covering every major staple crop on Earth. Now, here is what makes this story extraordinary. While Cho Han Kyu was watching Korean forest soil and figuring out how to bottle its biology, an entirely independent line of scientific research was unfolding 10,000 kilometers away inside a United States Department of Agriculture laboratory in Beltsville, Maryland. In 1941, 2 American researchers named J.W. Mitchell and M.R. Whitehead published a paper in the Botanical Gazette describing something they had observed about pollen. When they extracted compounds from corn pollen and applied them to other plant tissues, those tissues grew dramatically. The effect was reproducible. The mechanism was unknown. The paper was largely ignored for almost 30 years. Then, in 1970, Mitchell returned to the question. This time, he and his team extracted oily compounds from rapeseed pollen, the pollen of Brassica napus, and applied it to pinto bean seedlings. The growth response was not subtle. The stems elongated rapidly. Cell division accelerated. The compounds caused plants to grow in ways that auxin and gibberellin, the plant hormones known at the time, could not explain. They called the new family of compounds brassins. They published the finding in Nature, the same journal that published Darwin's most famous works. In 1979, after processing more than 500 pounds of bee-collected rapeseed pollen to extract just 4 milligrams of pure compound, a USDA team finally crystallized the most active member of the family. They called it brassinolide. X-ray crystallography revealed something stunning. Brassinolide was a steroid, a real, literal steroid molecule structurally similar to the steroid hormones in animal bodies. Plants had their own steroidal hormones. They had always had them, and no one in plant science had noticed for 200 years of research. Brassinosteroids became recognized as the 6th class of plant hormones. They are now known to control cell elongation, cell division, photosynthesis, vascular development, seed germination, fruit yield, and resistance to virtually every form of biotic and abiotic stress. They are active at concentrations of parts per billion. They are produced naturally in every plant on Earth in pollen, in seeds, in roots, and in young, growing tissue. Here is the part where the two stories collide. The microorganisms that Cho Han Kyu collected from Korean forest floors, the ones that multiply inside a bucket of Jadam Microbial Solution every 48 hours, are not just generic decomposers. A massive body of peer-reviewed research published in journals such as Frontiers in Plant Science, Plants, BMC Plant Biology, and the Journal of Plant Interactions has documented exactly what those microbes do. They produce hormones, specific bacterial genera that dominate forest soil and proliferate inside JMS buckets, including Bacillus, Pseudomonas, Azospirillum, Serratia, Stenotrophomonas, and Arthrobacter have been confirmed to manufacture plant phytohormones as part of their normal metabolism. A 2024 review titled Biosolutions for Green Agriculture, published in the National Library of Medicine database, lays out the chemistry in detail. These bacteria produce indole-3 acetic acid, the master auxin that controls root growth. They produce cytokinins, the hormones responsible for cell division and shoot multiplication. They produce gibberellins, the hormones that control stem elongation and seed germination. They produce ACC deaminase, an enzyme that breaks down ethylene and protects plants from stress. And here is the part that almost nobody outside specialist microbiology has heard. Some of these soil bacteria also produce compounds with brassinosteroid-like activity, the same family of plant steroids that USDA scientists spent 40 years trying to identify in plant tissue. The microbes were producing them all along. They had been doing it in every healthy forest floor on Earth for hundreds of millions of years before any scientists named a single molecule. Let me explain what this means in practical terms. When you brew JMS in a bucket, you're actually constructing a microbial bioreactor. You provide a starch source, the potato. You provide a mineral source and an osmotic stabilizer, the sea salt. You provide a population of mixed microorganisms, the forest leaf mold, and you provide water and an anaerobic environment, the bucket. Within 24 hours, the dominant aerobic bacteria, primarily Bacillus species, begin multiplying explosively. Within 48 hours, the population has doubled at least once, often multiple times depending on temperature. By 72 hours, the culture shifts into a facultative anaerobic state, where lactic acid bacteria and other fermentative organisms begin dominating. The starch from the potato is converted into simple sugars. The sugars feed the bacteria. The bacteria produce organic acids that solubilize minerals from the salt, and throughout this entire cascade, those bacteria synthesize phytohormones at a rate of hundreds of times what they would produce in static soil. When you pour the resulting liquid onto your garden, you're not just adding water. You are adding a hormonal cocktail that includes auxins, cytokinins, gibberellins, brassinosteroid analogs, vitamins, organic acids, and a population of live beneficial microbes that immediately begin colonizing your soil and continue producing hormones in situ for weeks afterward. There is no commercial product on Earth that delivers all of these compounds simultaneously at this concentration in a living, biologically active form. The reason there is no such product is not scientific. It is structural. A living bacterial culture cannot be bottled and shipped without continuous refrigeration, special packaging, and an aggressive expiration date. The chemistry that makes JMS so powerful is the same chemistry that makes it commercially impossible to mass produce as a shelf-stable product. Now, you might be wondering whether any of this has been formally tested, whether anyone has compared this homemade bucket brew against commercial alternatives in controlled trials. The answer is yes, and the results matter. Research published across multiple journals, including the journal Plants from MDPE, a Frontiers in Plant Science review on plant growth promoting rhizobacteria commercialization from 2018, and various Indonesian and South American agricultural studies have repeatedly shown that microbial brews containing diverse PGPR populations outperform isolated single-strain commercial inoculants. A Bacillus subtilis volatile organic compound study published by the National Library of Medicine found that volatile organic compounds released by this common soil bacterium increased tomato biomass, photosynthesis, and endogenous gibberellin, auxin, and cytokinin levels significantly compared to untreated controls. A separate trial on Pseudomonas fluorescens documented increased root architecture, leaf chlorophyll content, and yield in tomato and potato crops. The cumulative body of PGPR research now exceeds 10,000 peer-reviewed papers. The science of microbially produced plant hormones is not fringe. It is mainstream microbiology. What is fringe is the suggestion that a farmer can cultivate the same microbial community at home for the price of a 5-gallon bucket and a potato. That suggestion is heretical, not because the science contradicts it, the science fully supports it. The suggestion is heretical because it threatens the commercial model. Here is the question the industry hopes you do not ask. If indigenous microorganisms can be cultured at home, if the resulting brew demonstrably produces the same hormones that commercial products sell, if the entire process costs under $2 per batch and produces enough material to fertilize an entire garden, why is the global biofertilizer market worth over $3 billion and growing at 14% annually? The answer comes down to the same 3 compounding factors that suppressed willow water knowledge in the English-speaking gardening world for a century. First, standardization and shelf stability. A commercial microbial inoculant sold by a company like Verdesian Life Sciences, Bayer, Syngenta, or Corteva delivers a specific named strain at a specific concentration. Bacillus amyloleac faciens at 500 million CFUS per gram. Pseudomonas fluorescens at 100 million CFUS per milliliter. These are precise, repeatable, regulatory-approved formulations. A bucket of JMS contains a wild, mixed, ever-shifting population of organisms whose exact composition varies based on the forest soil used, the temperature, the season, the water source. Industrial farms cannot operate on that variability. Home gardeners can, and home gardeners benefit from biological diversity in a way that monoculture single-strain products cannot replicate, but the industry built itself around the more profitable, more controllable model and trained consumers to believe that diversity is somehow inferior to standardization. It is not. In ecology, diversity is resilience. Second, the appearance of complexity. The biofertilizer aisle of a garden center looks like advanced chemistry. The packaging uses scientific names, percentages, microscope imagery, certification stamps. The product looks like something that requires a PhD to formulate. A bucket of fermenting potato water does not look like that. It looks like something a peasant would make. It looks rural. It looks backward. And nobody builds a billion-dollar industry around a process that looks rural. But the science underneath the bucket is not less advanced. It is identical, the same microbes producing the same hormones, performing the same functions. What is different is the packaging, the marketing, and the margin. Third, the knowledge gap is linguistic and cultural. The Jadam website is in Korean. The original Cho Han Kyu books were written in Korean. The technique spread across Asia and Latin America for decades before any meaningful English translation existed. Search for IMO or JMS or microbial farming in Korean or Spanish or Portuguese, and you will find tens of thousands of detailed tutorials, video demonstrations, before and after photographs, community forums. Search for the same techniques in English 10 years ago, and you found almost nothing. The technique existed in a linguistic blind spot. Korean farmers had been culturing their own biofertilizers in 5-gallon buckets for 2 generations, while English-speaking gardeners were paying $50 for the same biology in a 1-liter jug imported from Germany. This is not unique to KNF and Jadam. The same pattern repeats with fermented plant juice, with fish amino acids, with lactic acid bacteria serum, with oriental herbal nutrient. Most of the regenerative agriculture toolkit was developed and refined in Korea, Japan, India, and the Philippines, decades before Western agriculture even recognized that soil biology mattered. The knowledge was never hidden. It was simply not translated. Now, after all of that context, here's the recipe. The 5-minute setup that starts a microbial colony doubling every 48 hours in a bucket. Pay attention because the details matter. You need a 5-gallon plastic bucket, just a standard bucket, the kind sold at hardware stores. Do not use metal. Metal interferes with the microbial activity and can corrode in the slightly acidic fermentation environment. You need 4 gallons of clean, non-chlorinated water. This is critical. Chlorinated tap water will kill the bacteria before they ever establish. If you only have chlorinated water, leave it in an open container in the sun for 24 hours to let the chlorine evaporate. Rainwater is best. Well, water is fine. Filtered water works. You need 1 pound of plain white potatoes, about 2 to 3 medium potatoes. Wash them, but do not peel them. Cut them into chunks and boil them in roughly half a gallon of water for 20 to 30 minutes until they are soft enough to mash easily with a fork. The starch from the potato is the carbohydrate fuel that the microbes will multiply on. The boiling water becomes a starch-rich broth that you will pour into the bucket along with the potato. You need approximately 2 tablespoons of unrefined sea salt, not table salt, not iodized salt, sea salt or rock salt, the kind with trace minerals still in it. The salt provides minerals, regulates osmotic pressure, and selectively favors the growth of the beneficial microbes over harmful ones at this concentration. You need 1 to 2 cups of leaf mold soil. This is the critical inoculant. Walk into the nearest undisturbed deciduous forest, push aside the top layer of fallen leaves, and dig down 2 to 4 inches into the dark, crumbly, fungal-rich soil underneath. That soil contains the entire microbial community you are about to multiply. If you cannot find forest soil, use the richest, oldest compost you have access to, or worm castings from an established worm bin. The assembly is the 5 minutes part. Take a pair of clean knee-high socks or a piece of cheesecloth. Place the boiled potatoes into 1 sock or cheesecloth pouch and tie it shut. Place the leaf mold soil into another. Hang both pouches inside the bucket so they are submerged in 4 gallons of non-chlorinated water. Pour in the sea salt. Stir everything together. Place the bucket somewhere shaded, ideally between 60 and 85 degrees Fahrenheit. Cover loosely with a lid or cloth to keep insects out and minimize evaporation, but do not seal it airtight. The fermentation will produce gas. That is the setup. 5 minutes of actual work. Now the biology takes over. Within 12 hours, the bacterial population begins multiplying. Within 24 hours, you will see foam forming on the surface. By 48 hours, the foam will be thick, dense, and frothy, sometimes bubbling visibly. This foam is the indicator that the colony has hit its first major exponential phase. By 72 hours, the brew is fully active and ready to use. In warmer climates, it can be ready in 48 hours. In cooler climates, it may take 4 days. The visible cue is the foam. When you see a thick, almost yeasty cap on top of the water, your JMS is alive. To use it, dilute the JMS at a ratio of 1 part JMS to 10 parts water. Apply it as a soil drench around the base of plants. Apply it once every 2 to 3 weeks during the growing season for routine soil health, or weekly during the active growth phase if you are trying to regenerate depleted soil. A single batch of JMS can also be used to start an unlimited number of new batches. Take half a cup of finished JMS and add it to a fresh bucket of water, potato, and salt. You skip the leaf mold step entirely after the first batch. The microbial colony is now self-perpetuating. This is the doubling forever feature. As long as you keep feeding the colony keeps multiplying. 1 bag of potatoes can produce an entire growing season's worth of biofertilizer from a single original inoculation. There are several ways to enhance the basic recipe. Many practitioners add a small handful of rice grains alongside the potatoes for additional starch and to introduce different bacterial strains. Others ferment the JMS first and then aerate it for the last 24 hours using a simple aquarium air pump, which shifts the microbial population toward more aerobic species and increases certain hormone yields. Cho Young-sang himself recommends using rainwater rather than tap water whenever possible, because the trace dissolved minerals in rainwater feed the microbial population more effectively than purified water. Some growers in tropical climates add a tablespoon of brown sugar or molasses to accelerate the initial bloom, though this is optional and not part of the original Jadam specification. The applications extend far beyond the home vegetable garden. In Korean commercial orchards, Jadam Microbial Solution is used as both a soil drench and a foliar spray on apple, pear, peach, and grape trees. In Filipino rice paddies, similar microbial brews are flooded directly into the standing water. In Argentine and Chilean vineyards, microbial inoculants in the Jadam tradition have been used for grape propagation and soil rebuilding after years of conventional chemical agriculture. In Tanzania, smallholder farmers use IMO-derived brews on cassava and maize as a complete replacement for synthetic nitrogen fertilizer. In urban gardens from Los Angeles to Berlin to Sydney, hobbyists run 2 or 3 buckets of Jadam Microbial Solution continuously through the growing season, treating every container plant, every raised bed, every fruit tree on their property. The transformation reported by long-term practitioners is consistent. Soils become darker, looser, more crumbly. Compacted clay becomes workable within a single season. Sandy soils retain water better. Plants resist disease more effectively. Yields increase, sometimes dramatically. Pest pressure declines. Most strikingly, the gardens become less dependent on inputs over time. The microbes establish, reproduce in the soil itself, and continue functioning without needing constant reapplication. The bucket is the starter culture for a permanent biological transformation of the land. I want to be honest about the limitations because credibility matters more than enthusiasm. JMS is not a silver bullet. It does not replace the need for compost, organic matter, mulch, or basic mineral inputs in severely depleted soils. The microbes need carbon to feed on, and soil without organic matter cannot host a robust microbial population, no matter how much JMS you pour onto it. The brew is also temperature sensitive. Below 60 degrees Fahrenheit, fermentation slows dramatically. In freezing temperatures, the active culture will die. Storage life of finished JMS is also short, typically 2 to 3 weeks at room temperature before the population shifts toward anaerobic putrefaction, and the brew develops an unpleasant smell. You make it, you use it, you make more. There is no shelf life because there is no shelf. Some plants, particularly very young seedlings, under 2 weeks old, can be damaged by undiluted JMS due to its acidity and microbial load. Always dilute. Always start with a lower concentration on new applications and observe the plant's response. And finally, The variability that makes JMS so powerful biologically is also its commercial weakness. 2 batches made by 2 different gardeners from 2 different forests will not be molecularly identical. Both will work. They will simply not be the same product. If you require laboratory-grade consistency for high-value commercial production, you may still need standardized commercial inoculants alongside your home brew. For ordinary home gardening, the variability is irrelevant. One safety note: sea salt is essential to the recipe, but it must be used in the specified ratio. Excessive salt will kill the microbes and damage your soil if applied repeatedly at high concentrations. Stick to the proportions. Always dilute at a ratio of 1:10 before application. Never use iodized table salt. Never use salt substitutes, and never pour undiluted JMS directly onto plants. And if you have any concerns about pathogenic bacteria in your starter material, particularly if you are using compost rather than forest soil, allow the full 72-hour fermentation. The lactic acid environment that develops during fermentation suppresses most pathogenic organisms, but do not shortcut the timeline. The market numbers complete the picture, just as they did with the willow water and lentil hormone story. The global biofertilizer market reached approximately $3.7 billion in 2024 and is projected to exceed $8 billion by 2030, growing at roughly 14% per year. The plant growth promoting rhizobacteria segment, specifically the bacteria producing hormones category we have been discussing, accounts for over 40% of this market. Major commercial players include Bayer, Corteva, Valent Biosciences, Novozymes, Lallemand, Verdezion, and a long tail of regional specialty firms. The margins on freeze-dried bacterial inoculants are extraordinary. A jar of standardized Bacillus subtilis selling for $25 at retail represents pennies of actual production cost. The remaining markup pays for packaging, marketing, regulatory compliance, distribution, and profit. Meanwhile, a 5-gallon bucket of JMS produces 40 gallons of finished diluted soil drench for under $2 in materials with microbes that include not just one strain, but dozens working together synergistically in ways that single-strain commercial products cannot replicate. This is not the only example. The same pattern appears across the entire input side of modern agriculture. Mycorrhizal fungi that occur freely in healthy forest soil are sold as packets. Trichoderma cultures that grow on rotting wood in any deciduous forest are extracted, freeze-dried, and bottled. Beneficial nematodes are farmed and shipped overnight in refrigerated boxes. Compost teas that anyone can brew in a bucket are sold as proprietary blends. The entire commercial model rests on convincing consumers that biology is too complex to produce themselves. That model has begun to crack as Korean natural farming, Jadam regenerative agriculture, and permaculture movements have spread across continents over the last decade. The cracks are spreading. The knowledge is finally crossing language barriers. Cho Han-kyu died in 2021 after spending 6 decades teaching farmers across more than 30 countries how to make their own inputs. He never patented a single technique. He never accepted royalties. He published everything. His son continues that work today through the Jadam organization, which operates as a nonprofit educational network rather than a commercial enterprise. The recipes are free, the lectures are free, the videos are free. They are translated into English, Spanish, Portuguese, French, Swahili, Tagalog, and a growing number of other languages. The knowledge is being released into the world deliberately against the gravitational pull of the commercial system that would prefer to keep it locked behind branded packaging. Here is what the biofertilizer industry never wanted you to understand. The most powerful soil amendment on Earth is not locked behind a patent, or a brand name. It is reproducing itself right now in every healthy forest floor on every continent. It can be collected with a shovel. It can be multiplied in a bucket. It can be applied for free. Chohan Q saw it in 1965. Mitchell and the United States Department of Agriculture team identified some of its molecular outputs in 1970. Microbiologists have spent the past 50 years documenting exactly which bacteria produce which hormones and in what concentrations. The science is complete, the biology is settled. The barrier between you and a thriving, regenerating, microbially rich garden is a single trip to a forest, a stop at the grocery store for a bag of potatoes, and 5 minutes with a 5-gallon bucket. The forest floor knows. Chohan Q knew. Mitchell and the United States Department of Agriculture team confirmed the chemistry. The peer-reviewed literature is settled. Now you know, too. If this story changed the way you look at the microscopic life in your soil, please subscribe and like the channel. We are uncovering the biological secrets that the industry has tried to monetize and hide. Let's grow a sustainable future together. The next treasure of nature's wisdom will be revealed soon.